So, now that we have RNA nucleotides, how can they be assembled into sequences? When RNA nucleotides dry on montrillonite clay or freeze on ice, they will polymerize into short chains. When conditions become wet again, most of these oligomers will break down into nucleotides. However, should some persist to the next drying cycle, there can be some template-driven polymerization where A binds with U and G binds with C, with a new strand being based off the first. When conditions become acidic, the hydrogen bonds binding the strands will become weaker, allowing the strands to separate. Furthermore, should hairpin loops form, the strand will loop around and will get longer as free-floating nucleotides pair up with nucleotides further downstream, allowing for template-driven elongation to occur. However, some problems are that non-complementary base pairing will be common, and there will be a mix of phosphate groups binding to the first and third carbon of the adjacent nucleotides. At this point, we have RNA sequences floating around the environment, but could they carry out catalytic activity? While the odds of any one sequence having a catalytic function are small, of the myriad of sequences produced, odds are some could facilitate early metabolic reactions. With many short sequences being produced via wet, dry, and acidic basic cycles, a likely function of an early ribozyme would have been a ligase, combining the assorted oligomers into larger polymers. This would be the first form of self-replication. Because of the sequences that a ligase would assemble, some of them would be made of its own constituent parts, so it would in essence replicate itself when it assembled a new ligase ribozyme. Furthermore, complementary base pairing between parts of the ribozyme and free-floating RNA oligomers would make it more likely that early ligase ribozymes would bind with RNA sequences more closely related to themselves, allowing for selective pressure to act on mutations that gave the ligases greater selectivity. Later on, a ribozyme that would assemble RNA sequences using a template strand and free-floating nucleotides, or a polymerase, could evolve. As of now, no polymerase ribozyme has been developed that can replicate a sequence greater than or equal to its own length. But once polymerases have produced new sequences, they can be combined by the aforementioned ligases. Polymerization requires activated nucleotides. The nucleotides with one phosphate won't cut it. Modern polymerization reactions use triphosphate nucleotides. When these are combined, the second and third phosphates are released. However, it has been experimentally demonstrated that diphosphate nucleotides can be used in polymerization reactions. Each RNA oligomer that makes up these ribozymes will have a corresponding template strand, which the polymerases use to assemble the oligomer. And the formation of tertiary structures like hairpin loops and folds implies that the polymerase ribozymes will more often carry out polymerization reactions on the template strand. This would lead to a situation where sequences incorporated into ribozymes are produced far more quickly than their source strands, giving rise to a system reminiscent of modern molecular biology, where a gene is transcribed far more often than it is replicated, with the template strand acting as DNA and the strand incorporated into the ribozyme acting like the mRNA and protein. Apart from replicating themselves, there are other potential functions ribozymes could have carried out. Whereas enzymes catalyze reactions via interactions between the amino acid R group and the substrate, ribozymes do so via interactions between the nitrogenous bases and substrate. Often, an enzyme or ribozyme can utilize several different chemically similar substrates as a result of mutations, although with varying degrees of affinity for each. But should utilizing a minor substrate suddenly become advantageous, the ribozyme will then evolve to have a greater specificity for it until said substrate becomes the major substrate. Given that polymerization reactions involve transferring phosphate groups between carbon atoms, an early function of ribozymes was likely reacting reactive phosphorus species with organic molecules to phosphorylate them so they could be fed into prebiotic metabolic pathways, such as the abiotic PPP analog. Another early function of ribozymes might have been catalyzing certain steps of nucleotide synthesis, such as converting adenine to guanine and uracilcytosine. Ribozymes can evolve to catalyze metabolic pathways through incremental stages. It starts with the ribozyme catalyzing the first step of a pathway until a mutation happens, allowing it to convert the first product into the second, which is essentially a slight tweak because the ribozyme already has a binding site that the first product can fit into. This process repeats itself until the entire pathway is facilitated by ribozymes. These ribozymes would have this phylogeny depicted here, with the ribozyme catalyzing the first stage in the pathway being the outgroup, and the ones catalyzing the last and second to last being an in-group. However, if a family of ribozymes evolved to synthesize nucleotides, it likely would have evolved from a polymerase ribozyme, which takes nucleotides, the end product, as a substrate. So, the first nucleotide synthesizing ribozymes would catalyze the final steps of nucleotide synthesis, meaning the phylogeny would have been flipped horizontally. While these reactions can proceed in an abiotic environment, adding a biocatalyst would increase the rate, limit the number of products that can't be used, and produce compounds with the correct stereochemistry. However, one distinction that I think should be made is that saying that early life had ribozymes is not necessarily affirming the RNA world hypothesis. This is because the RNA world hypothesis postulates that ribozymes catalyzed all biotic reactions before the advent of enzymes, and what we are finding is that many of these reactions were likely facilitated by inorganic mineral catalysts. While the notion of an RNA genome can resolve many conundrums about the origin of life, there are a number of problems. One is that all polymerase ribozymes produced thus far have a low fidelity rate, with 8% of all nucleotide insertions being mismatched mutations. Another problem is that RNA polymers are fairly unstable because of the steric hindrance from the hydroxyl group on the second carbon and may break down faster than replication is possible. A third contention is that abiotically produced nucleotides will have a 50-50 mix of the D and L stereoisomers ribose, and a sequence would have to be made up of entirely one or the other, with the presence of the wrong stereoisomer having an inhibitory effect. However, UV light will selectively break down the L stereoisomer, and the D stereoisomer is used by life today. Another problem is that we have not yet determined a way activated nucleotides could be produced abiotically, although reactive phosphorus species from tribosite open some possibilities. Finally, it has been argued that the probability of catalytic RNA sequences emerging from random chance is quite low. While it has been known that scrambled protein sequences can evolve novel functions, proteins have 20 amino acids, while ribozymes only have 4 nucleotides, limiting their catalytic repertoire. Given the problems associated with an RNA genome, some have hypothesized that other genetic materials were used by the first organisms. Pyranosol, or pRNA, uses a 6-carbon sugar known as pyranose instead of ribose, and it is more stable and easier to replicate. Tetrose nucleic acid, or TNA, uses a 4-carbon sugar that is again more stable and can easily be produced on the prebiotic Earth, and this will base pair with RNA. This means that it could act as a stable genome that would be used as a template for the polymerization of ribozymal oligomers to be used as ribozymes. Similarly, peptide nucleic acid, or PNA, which has a peptide backbone, will base pair with RNA. Anelyl nucleic acid, or ANA, can incorporate both stereoisomers into a stable sequence. Finally, thioester peptide nucleic acid, or tPNA, can spontaneously assemble into a sequence on a template and is very stable. Some have even proposed that the first life didn't possess any genetic material and instead passed information from one generation to the next via their chemical constitution or their composome. However, computer simulations have shown that this is highly unlikely. Now that we've covered the origin of metabolism and replication, let's discuss the origin of compartmentalization. Modern cells have at least one plasma membrane surrounding themselves to keep their internal components inside. This membrane is also used to control what goes in and out of the cell and maintain a cross-membrane salinity or pH gradient. Most membranes are around 10 nanometers thick and are composed of lipids, the majority of which are phospholipids. These lipids have a charged hydrophilic head group and a pair of nonpolar hydrophobic tails, making them amphiphilic. This means that when they assemble into a membrane, the head groups will be on the outside of the membrane and the tail groups inside. We may think of these membranes as solid structures, but they're actually fluid mosaics and have many different kinds of molecules, such as enzymes and receptor proteins embedded within them. These molecules move about freely as if they were in a liquid, except in two dimensions. Apart from having a membrane surrounding the cells, eukaryotic organisms have membrane-bound organelles, and most prokaryotes have two membranes surrounding their cells. In order to get lipid membranes, there must first be lipids. In the modern environment, Fischer-Tropsch type reactions occur in hydrothermal systems between 150 and 250 degrees Celsius. This reaction proceeds in a manner quite similar to the aforementioned production of alkanes, but sometimes an alkane chain is bound to a carboxyl group that is also attached to a transition metal atom and then released as a carboxylic or fatty acid. Trace amounts of cobalt in the metal catalyst facilitate the transfer of methyl groups to adjacent alkane chains, allowing the hydrocarbon tail to become much longer than it could otherwise. Vitamin B12 is used in methyltransferase reactions in modern biological systems, and it contains a cobalt ion. Although montmorillonite clay can't facilitate the production of lipids from carbon dioxide or bicarbonate, it can catalyze the production of lipids from much smaller organic acids and volatile hydrocarbons such as formate, ethane, and ethylene.